In a world where space and time are intertwined, one man dared to challenge the status quo. His name? Dayton Miller. His weapon of choice? A flute. But not just any flute. This instrument held the power to unleash the ether, a pervasive medium once thought to fill the universe. Miller, a physicist by trade, a musician at heart, discovered a harmonic resonance within the ether. He found that his flute, when played with precision, could manipulate this elusive medium. His melody.
Okay.
All right, hello and welcome to Ether Cosmology Roundtable number 70. Today we're joined by Taylor and her husband Dan. They're going to be doing a presentation on abiogenesis and how it's linked to evolution as part of the microevolution, proving the macro, right? So she's going to go through a systematic dismantling of that. And whenever you guys are ready.
So I'm just wondering, can you see both of us on camera? Because right now I'm just looking at my presentation, so I can't see what it looks like.
Pretty
You
good.
see the
yeah.
right side of Dan's face, yeah.
Yeah, we can see
Yeah,
most of
both
Dan.
you, yeah.
There we go.
Okay,
Both of you
better?
slide to the right or pan it to the left. Yeah.
There you go. Nice.
What about now?
Perfect.
Yeah, you're in there, sure.
Okay, cool. So today I'm going to be presenting on does science suggest abiogenesis or intelligent design? All right, so this is an introduction to biology. Biology is the study of life and living organisms, and cell biology is the study of cells. Cells are basically the fundamental unit of life, and you can look at the tree of all the different branches of biology over here, including cell biology, which is what I study right here in the middle. And a little fun fact is that the cell is derived from the Latin term cellula, meaning small room. All right, so I'm going to go over some of the fundamentals of biology just so you guys can kind of follow along with the presentation, give you guys a good introduction so you'll be able to understand what I'm talking about. All right, so biology has a hierarchy called the biological hierarchy, where basically you start at an atom and you go up to a molecule. Molecules make up organelles, organelles make up cells, cells make up tissues and organs and organisms, ecosystems, and eventually the biosphere. So there are some characteristics of life. All living things are made of one or more cells. They display organization, they can grow and develop, reproduce, respond to stimuli, they require energy, and maintain homeostasis, which is kind of like equilibrium. All right, so, sorry. All right, so I'm going to give you guys some cool microscopic views of some single-celled organisms and individual cells, human cells, etc., just to kind of show you what biologists are looking at, what they're studying, and what life looks like on the micro scale. All right, so over here, here's the dancing diatoms, the Bacillera pexifera, and they're really cool.
Taylor,
Are you
Yeah.
sharing the same slide?
Yeah, Taylor, 1 second. Your screen share is stuck on the first slide.
Oh, no.
Yeah, I think you need to reshare.
Can you cut off the screen share?
Are you screen sharing in full screen mode?
I'm not sure.
You just
No, but-
needed to click slide 2 in the margin on the left.
Okay, what about now? Can y'all see it moving?
Click on slide 2. Still looks like
no.
slide 1.
Yeah.
Yep, there we go. You're good now. Now it
Is it
works.
showing the full?
When
Yep.
I push play,
It's the full thing.
go ahead and push play. Like,
What if you
full
go
play.
full screen? Would that
Play
be
from
better?
start. Do y'all see my whole slideshow whenever I leave full screen?
Like, what about now? Is it
No.
changed now?
No, we
Yeah,
only
we don't see full
see
screen.
we don't see the full screen.
Yeah, I think so. You're sharing application. Full screen would mean we share a window, I think.
I think it's being. I guess the whole is the slide lighting up the whole like window.
No. So what you should
No.
do is go into share your screen on Discord and click share entire screen and share that. Then when you go into full screen, we'll see the whole thing.
Yep.
How do you do
I
that?
don't see share your entire.
So change. You go into the
Window.
share your screen button there in Discord, and then just like change it.
Check
I
the
see Mount
tab.
Wilson, I see my presentation, and then window.
entire screen.
Yep,
Okay,
so-
cool.
He's got it.
I'll now start it. Make it. Okay, now what about now?
You got it. There we go.
There we
Now
go.
we're good.
Nice.
All right.
Perfect. Okay, we'll just start it over then. And I'll just
Nice.
skim through it real quick.
Cool.
Yeah, so
We lost you on camera. though,
biology.
if you want to bring
Okay,
camera
are you
back.
good?
Yeah,
Y'all
yeah,
can't see me anymore now?
No,
Oh no.
Can't see you guys.
Okay, let me try it again.
Yep.
Okay, what about now?
back.
100% good.
Okay, perfect. And I don't know if I'm going to be able to fit me and Dan in the whole presentation, so because I'm like having to look, like some of the words
Yeah.
are very tiny.
You're good.
All
It's
right,
close
so-
enough.
Okay, so does science suggest abiogenesis or intelligent design? All right, let me go to next. All right, so as I just mentioned, biology is the study of life and living organisms. I study cell biology, and cell biology is the study of cells. And one fun fact is that the word cell is derived from the Latin term cellula, meaning small room. And cell biology is essentially the study of cells. Yeah, I mentioned that. Yes. Okay. So now the fundamentals of biology, right? Biology has a hierarchy called the biological hierarchy. So you start at an atom and you move up to molecules, organelles, cells, tissues, organs, and so on and so forth until you get to the biosphere of the Earth. And then here are the characteristics of life on the right side. And so all living things need to be able to have these characteristics. Okay, so right here is a single-celled organism called Bacillera paxifera, and it's also called the dancing diatoms. So they basically slide back and forth upon one another. And here's a picture I took of them. So I'm basically just trying to show in this slide just the complexity and amazingness of these single-celled organisms. Just because something is a single cell does not mean it's like not complex or simple. These are very intelligent, very sophisticated, etc. Okay, here's another picture of a protist here on the right, and then here's some cool looking diatoms in the middle that were designed by Klaus Kemp. He basically put them in this nice little order and made artwork out of them. And so he designed that, and I believe that the diatoms are also intelligently designed, and I'm going to be making my case for that. All right, here's some more videos of single-celled organisms. Here is basically a cell dividing. And over here on the right is a macrophage looking for a little bacteria. And then on the left, here's some neurons making some connections. And then here's personally what I study, microtubules. In the middle, you can see an animation of a microtubule forming. And I'm tracking this real time via fluorescent microscopy. And I'm looking at Clanomonas reinhardti, which is my model organism. And there's a fun fact that microtubules are attributed to fiber optic cables and neurons. All right, so now we're going to start looking at some intercellular components of the cell. So cells have these basically subcomponents called organelles, and they're present in all cells. So some different components are the lysosomes, the vesicles. You got the cytoplasm, which kind of fills up the entire cell. The nucleus, which houses the DNA. You got chromatin, which basically is holding the DNA around in protein. Basically, it's like these proteins that the DNA is wrapped around. And then you have the rough endoplasmic reticulum, smooth ER, and so on and so forth. And you can actually look at these organelles using an electron microscope. So right here is the Golgi apparatus, and you can actually look at the Golgi right here in the cell diagram just to see like kind of where we're getting this from, right? We got the mitochondria we can see right here in the cell, and so on and so forth. And here's a fun fact. Dr. McGill, he's the director of molecular visualization at Harvard Medical School, and he composed this image in the top right. And he said, although people are struck by the complexity and density of this environment, what we depict is actually a very dilute in its concentration relative to a real cell. So just think about that for comparison. This is a dilute concentration of what it looks like in a cell. So that's how complex it is. All right, so I'm going to go over cellular composition. So basically, cells have these 4 macromolecules. We have the proteins, lipids, carbohydrates, and DNA and RNA. So proteins are made up of amino acids, lipids are made up of fatty acids, carbohydrates are made up of sugars, and then DNA and RNA is made up of nucleobases or nucleotides. And biological molecules exhibit something called chirality, meaning that they cannot be superimposed on top of one another. So you can look at this depiction of this hand right here to see what chirality actually looks like, right? When you can't superimpose them. So when I superimpose my hands, they don't perfectly align over one another. Okay, so homochirality in the cells provides them with integrity and preserves function. You might have to shut there. Okay, so proteins are left-handed, lipids are mostly right-handed, carbohydrates are mostly right-handed, and then DNA is also right-handed, meaning they're all aligning in that fashion. All right, so here's a little fun facts page on the molecular composition of the cells. So the smallest cell is made up of 100 billion atoms on estimate, on average. So then they saw how many proteins were in a yeast cell, and it was 42 million. And the bacteria with the shortest DNA, Mycoplasma, has 507 genes and 580,000 base pairs, which would be around 34 million atoms. So you can just see like how, like, even though they're small, they're very complex and not just easy to make. And these organic chemical reactions, they're positing.
And this large number of genes and base pairs for all those genes would be necessary. Right?
Yeah,
Like
these are necessary.
It's not just choosing to have so many genes, like it's all the different functions it needs to fulfill.
Exactly. And at the bottom right here is a fun fact about this bacteria. They basically cooked up in the lab, or they basically made a GMO and was messing with their DNA sequence. They were trying to see how small of a DNA sequence can they make and basically cut out portions of it for it still to survive, right? And the smallest DNA that they could get is a minimum of 438 protein coding genes and 531,000 base pairs. So even whenever they were trying to make like the simplest cell, it still required all these different genes, right? All this long, extremely complex strand of DNA. So yeah, even the most minimal life forms are extremely complex. And just, I want you guys to understand that. All right, and then over here, all biological organisms contain oxygen, carbon, hydrogen, nitrogen, phosphorus, etc. So these are required for all life. All right, so now we're going to go into cell functionality. So how does the cell work together? Where does it store information? And how does it use that information that is stored? All right, so here's the cellular information system. It's called the central dogma of biology, right? So it's basically the flow of information from DNA, where it basically stores that information, into a messenger RNA, which then basically takes that DNA code and then moves it out into the cytoplasm to the ribosomes to get transcribed to a protein. So it goes from DNA to RNA to proteins. And make sure you guys remember that, because I'm going to go over it a little bit later. And then a fun fact with the central dogma, it was Francis Crick coined the term, and dogma was actually a misnomer, as it implies an unchallengeable belief which contradicts the scientific method. And Crick himself later acknowledged this. So he should not have used the word dogma, because all science should be it's not like unchallengeable. You should be able to challenge science. And then right here is just a picture of E. coli DNA. All right, and so here's a little short video just describing the central dogma so you guys can understand. And it's about, I think, 2 minutes long. I'm gonna try to speed it up a little bit if I can. Let's see.
The DNA double helix contains two linear sequences of the letters A, C, G, and T.
Oops. Sorry.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make.
Can you mute your mic, Taylor, while it's playing?
Double helix and copying one of the two strands. The yellow chain speaking out at the top is a closed chemical cousin of DNA. The building blocks to make the RNA enter through an intake hole. They are mapped to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated to a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called the ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
The grid you can see in this microarray slide.
Gotta unmute your mic there, Taylor.
Sorry, can y'all hear me now?
Yep, welcome back.
Okay.
Welcome back.
Cool. Yeah. So now I'm going to be going over the origin of life. That was just kind of like an introduction, so you'll understand some of the concepts I'm going to be going over. All right, so the origin of life, was it just a random cosmic coincidence? So the scientific community hypothesizes that life formed through a random undirected process known as abiogenesis. Abiogenesis is basically the idea that life arose from non-living material. All right, and then here's a little fun fact. Panspermia is the concept that life may have originated elsewhere in the universe and then reached Earth through space travel. And Watson and Crick, who actually discovered the double helix of DNA, said that this must be intelligently designed and therefore it must be coming from some extraterrestrial planet. So basically, they knew the DNA was intelligently designed, but they were atheist. So they wanted to just basically push back the question and say, well, it could have been like some, extraterrestrial source, right? Maybe aliens or whatever. So anyway, it's just a little fun fact. But
Not
panspermia
God though, right? Not God though,
just not
right?
Just God.
Okay.
So yeah, so this really like begs the question, like when people say, "Oh, well, it could have been coming from another planet." Just know that that's not answering the question of how life originally formed anywhere in the universe, in any universe, for whoever believes that. All right. So here's oh my gosh, I just unplugged my. Okay, no, like I think the cord. I think the
EMP?
cord. I'm so sorry.
You're
I
good.
really. Yeah. Let me turn this back on.
Got
All
EMP?
right.
Oh, I see.
Cool.
I see
That's
now.
not a problem. Your camera's auto gain. It didn't even notice you turned it off. Don't worry about it.
Oh, really? Okay, cool. Perfect. Okay. So here's the history of abiogenesis, right? In 1859, Darwin proposed the theory of evolution in his book On the Origin of Species. And then in 1870, the term abiogenesis was coined by Thomas Huxley to describe the process by which life could arise from non-living matter. In 1871, Darwin described the possibility of life emerging in a warm little pond. And in 1929, Oprah and Halide proposed that life originated from a primordial soup, which is a mixture of organic compounds in Earth's oceans, energized by lightning or UV light. Okay, and over here is kind of a schematic of abiogenesis and how life could have formed, right? So you got basically this RNA being formed, turned into DNA, turned into cells, and then we evolved, and then now we have brains, and then so on, whatever.
I'm not buying it.
Yeah. All right, we're gonna test it. Hold on. All right, so I just wanted to give you guys a reference point. Whenever they came up with this theory of what they actually thought about the cell at the time, right? So here's a little excerpt from the Fortnightly Review, which was a very popular magazine in the 19th century, and it was written by George Lewis. And this was titled Mr. Darwin's Hypothesis. And in it, he was basically stating that the simplest form of an organism, organic life, is not as commonly stated a cell, but a microscopic lump of jelly or protoplasm. And the definition of a cell is a nucleus with surrounding protoplasm. So this just kind of helps you understand like what they actually thought of the cell at the time. And I'm going to get into how we started learning more about it. But yeah, so whenever they came up with this theory, they really had no idea about the complexity and the intricacies of even the most simple life form. All right, so here's abiogenesis continued. It was 70 years after abiogenesis was proposed that we could actually see the intricacies of the cell, right? So over here is some images from an electron microscope of different cellular components. They are very interesting. Here's the mitochondria, and then you got the chloroplast, and then here's the cilia, which the microtubules protrude out of. So it was in 1940 that Manfred von Andren constructed an electron microscope which greatly improved our ability to visualize cellular structures. And in 1959, the first electron micrograph of an intact cell revealed these subcellular structures.
Just one thing I want to mention about the cilia, like at the top, the thing that looks like a wheel.
Yeah.
If any one of these components of the cilia are not present, like you can genetically modify them so that they are missing little pieces, little connectors or stators, I think these are called, it doesn't function. And so I don't know the total number of proteins that are in here, but if any one of them are missing, it doesn't function, like I said. So that's an example of a irreducible complexity.
Right. Yeah. So if any part, irreducible complexity is basically if any part of it was removed, it would not function. So we're going to go over that in a little bit. Yeah. Okay. So I was trying to really steel man the abiogenesis position. in the current knowledge and theory on it. So I went and I was searching out what is the most commonly used biology textbooks. Okay. And then I found this Campbell biology book by Lisa etc. So basically, this was like the only page on abiogenesis that I could find. And it was basically just referring to the Miller-Urey experiment, which is what we're going to go over next. Okay. So here's the Miller-Urey experiment. It is like the most cited research done, the most cited experiment for abiogenesis to say, yes, this must be true. Okay. So this was in 1953. So here was the steps of his experiment. So he was trying to simulate Earth's early Earth's conditions, and basically he mixed gases, methane, ammonia, and hydrogen in a sealed chamber. He added water and heat to create the vapor, as you see over here on the right in the diagram. And then he added these electrical sparks representing lightning. And then he cooled and condensed the vapor, simulating rain. And then he collected and analyzed the tar that was produced. Okay, so over here on the right is what the result of his experiment would have came out with. So it basically just looks like a dirty bong, right? Let's be honest. So yeah, this is like yeah, one of the most cited experiments. And let's go over his
Can I ask
results.
you a question on that just before you move on?
Yeah, go ahead.
Okay, go back to that. Electrodes in the top center, 12 o'clock position of the diagram. What were his electrodes made of where he's doing the spark gap? I mean, that's got to be crucial to producing
so
this tar here, right?
Yeah, to make this tar, right. So this was only a 2-page manuscript. So it was like really lacking in the detail that a current peer-reviewed published paper would have. And you'll see that in the results section, which I'm about to go over next.
One additional question. Back to 1921 and 29 on the previous slide where they were talking about the two possibles.
Sorry.
There you go
one?
with Luz. The two possibles for the creation of abiogenesis in the first place being either lightning or UV light back in 1929. If I'm not mistaken, that's what you said.
Yeah, you're correct. So they basically assumed that they would have this like organic pond, and then somehow it would get struck by lightning, and then it would need to go through this like aqueous phase and then drying phase and stuff like that.
Okay, this may seem like a weird question, but these are scientists who proposed that?
Absolutely. And look, they're still cited in
Okay,
this text.
so how long was it until either one of them or some of their offspring, scientific offspring, actually replicated that in the lab with lightning or UV light? Who has created life from non-life in the lab using those two methods?
Nobody has created life from non-life ever, but they have been.
I just thought I'd check.
Yeah, they've never created life in the lab, and I'm going to show you how minuscule this result actually is.
And the electrodes.
Oh, for sure.
The electrodes were made of tungsten.
It was a loaded. Oh, tungsten. Very good. Thank you. No, it was a totally loaded question. I knew the answer, man. Nobody ever. So continue.
Yeah, if anybody has questions, I'd love to interact and answer them. All right. So yeah, we got this tar-like substance right here. And I don't even know how they got this stuff out of this flask. I would assume that they would just have to throw it away because it just looks destroyed. Anyway, it's like this really sticky hydrophobic substance that came out of the experiment. And that's, what we came from, right? All right. So here is the only results graph that was published. They used paper chromatography, and he was able to isolate aspartic acid, glycine, and alpha alanine and beta alanine. Okay, and so basically, down here is the chemical reaction that would have occurred. Right, you have the methane, ammonia, and hydrogen, and then he sparked it, and then he did like a heating and cooling type of reaction. And so here's what came out of it. We got the glycine, the beta alanine, and alanine, right? And so this, I mean, you can kind of predict like what's going to happen with these experiments, right? If you just mix these like CH4, NH3, H2O, and you subject them like to a high spark of electricity, they're going to form some weird things, right? So it's not that surprising that these amino acids came out of it. Right here is glycine at the bottom. It's only 10 atoms. Beta alanine's 13, and alanine was 13 atoms. And yep, it was basically just a fancy organic chemistry experiment. And yeah, so they found amino acids, and the amino acids actually only composed of 2% of the solid residue that came out of it. So only 2% of it was amino acids, and some people say this is an issue because if they were like bound up in this tar-like substance or that was around them, then those could either react with each other or stabilize each other or prevent them from basically going into the environment and reacting with other things that would have been necessary to continue on the process towards life. So another thing to mention, remember when I mentioned chirality earlier? Well, it is really important that all amino acids are left-handed. So this produced a result of left and right-handed amino acids. So even if we had theoretically these conditions and we had done this experiment, they wouldn't have been able to form a protein from this because they're left and right-handed and they cannot bond like that. You would need all left-handed basically to go through and do the chain. All right. And then in comparison, there's 100 billion atoms in the smallest cell. And this was this produced 10 atoms, 13 and 13, just to kind of give you a scale here. All right, so Dr. Scott Hughes actually pointed out some flaws in the Miller-Urey experiment. So here was a couple of the flaws that he pointed out. He says like the methane and ammonia were carefully selected to ensure the production of organic molecules, basically implying that this wouldn't have been like plausible in an earlier condition, right? They were carefully selecting everything. There's no evidence to indicate the Earth's atmosphere was reducing. He says a reducing atmosphere would be fatal. Lightning would have destroyed any organics. Molecules produced in the Miller-Urey apparatus would be detrimental to life forms. They basically designed apparatus to separate the amino acids from the mix. So after it was all finished, they like kind of separate them. That's what would have to happen in any consecutive steps towards life. Amino acids were all left and right-handed, as I mentioned, and then formaldehyde and cyanide were destructive to life and were present. So I understand there's a couple of these that can be contested, but overall, here's just some criticism that Miller-Urey received for his experiment. All right, so does the Miller-Urey experiment imitate early Earth conditions? So here's some quotes from some peer-reviewed papers that I saw. So it is possible that the primitive atmosphere was not as reducing as the Oparin, Urey, and Miller believed, right? And so also the accepted picture of the early Earth's atmosphere has changed. It was probably oxygen-rich with some nitrogen, a less reactive mixture than Miller, or it might have been composed largely of carbon dioxide, which would greatly deter the development of organic compounds. And so models suggest that there were significantly low amounts of NH3 concentrations in the ocean, unlike what they would have predicted.
I just want to mention that this high amount of carbon dioxide creates carbonic acid, and then it breaks down into bicarb and hydrogen ions. So it becomes more acidic, which produces an oxidizing environment rather than a reducing environment necessary for any of these experiments simulating the Miller.
Yeah, like for protein synthesis, etc. Like you need a reducing environment. So basically, he was putting them in the optimal conditions. But what the early Earth's atmosphere was, or what the conditions were, like, is very up for debate. So it's not really set in stone. Moving on. All right, so this is a continuation. Did Miller-Urey imitate early Earth's conditions? So in Urey's book, another thing is that they actually predicted that the oceans would have been like 10% solution of organic compounds. Okay, this would be a very high percent of solution of organic compounds. So current organic matter content from ocean sediment is approximately 1 to 3% of organic matter, right? So that's much lower than their 10%. And actually, we have life in the ocean now. Like, we have abundant life. So if we have 1 to 3% organic matter with this abundant life, like, it's just crazy that they predicted 10%. But I think they also thought the Earth would have much less water back then. But now scientists are saying that we have had a water world early Earth. So it's up for debate, but either way, it doesn't seem like in all cases he was trying to accurately represent the early Earth, rather than trying to like do like circular reasoning, saying, oh well, this is needed for life, therefore that's what it must have been. And other scientists also expressed that criticism. All right, so now I'm moving on to trying to figure out what the current abiogenesis model is. Right. So I went and looked through a bunch of abiogenesis papers. I found this one. It was only published like a few months ago. So what they were doing here is not as important. Basically, I was just using the paper to look at their introduction and discussion session to see where we're at in abiogenesis. Normally in an introduction to a paper, you're going to give like the current state of knowledge in the field of whatever you're writing about, and then give an introduction to your experiment. So anyway, this paper was basically just trying to use a computer code to infer the order of the addition of amino acids. All right, so here were some spots that I highlighted with it. So it's very interesting how many times they invoke products or whatever coming from extraterrestrial sources. So I'm going to give you one quote. So it says, the modern genetic code was likely assembled in stages, hypothesized to begin with early amino acids present on the Earth before the emergence of life, possibly delivered by extraterrestrial sources such as asteroids or comets. And like I was thinking maybe this is a one-off or like this isn't very common, but it is extremely common. Like for example, like when I was looking at the current research of lipid formation in prebiotic Earth, basically they were using a compound that came from meteorites. Like they were saying, oh, well, this compound's on meteorites, therefore that's what I'm going to use for the reaction. Like, so they're like almost having to invoke it. Like they're having to invoke these meteorites and comets to make some of the parts of their theory work. All right.
Can I ask a chemistry question right about there?
Go ahead.
When they talk about things coming in via asteroids or comets, how many of them address the absolute bitter cold of, quote, space, and that most of these chains and frankly anything containing water coming back up above 4C from a non-freezing state, anything melting from there, of course, would completely destroy its cellular form? Did they ever handle that?
Not really, but yeah, that's definitely something that they should be considering. But I can probably give you one of their responses, which is that, oh, well, it was bound up in rock, and therefore when it entered the atmosphere, it stayed stable, and then only after it crashed into the earth and got dispersed in water did it, whatever. But normally things that are bound up in rock disperse.
So it spent umpteen years at 2.3 Kelvin, and then it blew through the atmosphere at some ungodly speed, pun intended, and somehow, didn't burn up on its way through the atmosphere. So we assume that it only heated to a couple of 100 degrees, not actually melting point for the rock itself, and somehow life survived. All of that. Okay, next.
It's really sad that they even have to invoke this, but it is what it is. All right, so then I was looking at some of the other environments. So cyanosulfic environments and alkaline hydrothermal vents are also invoked a lot. So here's another statement on that. So amino acid abundances obtained from asteroid samples returns will also soon contribute. So they're all waiting on these asteroid samples.
Just as soon as they find one.
Right. All right. So then they also invoke the water rock interface because in many biotic reactions, you're going to have these like wet and dry cycles. So that also complicates the problem. And then this one's the funniest. Euclidean's subsurface ocean. Guess what that is? That is Saturn's moon. That is Saturn's moon. So yeah. And then hydrothermal vents again, and then tRNA synthetase. This is an interesting one that Dan's going to talk about later. So I'm going to give you this quote from her. It says, perhaps the biggest mystery is how sequences such as the common ancestor of basically Luca tRNA synthetase, which were translated via alternative or incomplete genetic codes, ended up being recorded or recoded for translation by the direct ancestor of the canonical genetic code. It's like, how are these things transferred over, like from this ancient, genetic code to what it is now? So that's interesting. But anyway, we'll go over that in a little bit. All right, so I read a bunch of papers on abiogenesis and was realized shortly after that they are having to invoke all of these different conditions for different types of reactions. So I wanted to make this little diagram. This is just a few of them, guys. Like there are so many more, right? So some of the things that I came across were these wet-dry cycles that were needed. They invoked lightning or UV light, clay, comets or meteorites, hydrothermal vents, shallow pools, porous rocks with subsections of environments within them, and crystals to help solve the chirality problem. All right, so they invoked this montromorillonite clay. That's for RNA polymerization, and that would have had to happen in tidal pools near some volcanic ash. Then we have pyrite, which is for pyrophosphate synthesis. Then we have these iron sulfur minerals for amino acids and nucleotide synthesis. And then those would have had to been in a hydrothermal vent. Then we have these borate minerals, which are needed for ribose stabilization. So you got this nice little hydrothermal vent pond with all these different components to fix all these different problems. And it gets worse because some of these cannot react in the presence of others. And I have that in the next slide. Okay, so here's the compatibility paradox. When you need each other, but you cannot coexist. Okay, so for DNA synthesis, right, we would need UV radiation and heat. We would need an aqueous environment and shallow ponds. Whereas for RNA synthesis, we would need UV radiation, heat, wet, dry cycles, and tidal ponds, geothermal areas, etc. And then for protein synthesis, we would need heat, drying cycles greater than 100 degrees, which contradicts with the DNA and RNA synthesis. Low water activity, which contradicts with the DNA synthesis and partly the RNA synthesis. And then these hot volcanic surfaces or evaporating pools. So you got the ponds over here, and then you also have like the hydrothermal vents for different conditions. So yeah, they would have had to form in different areas, and they say maybe in these porous rocks they can have these like subsections. So it's very interesting. So also like DNA would need like UV radiation would be needed for DNA and RNA synthesis. However, they did not need UV radiation for protein synthesis. And this is why one of the reasons why they say DNA and RNA would happen in the ponds, like on the shore or whatever, and then protein synthesis would happen in a deep sea hydrothermal vent. So yeah. And this is because whenever they try to form these molecules in the lab, they have different conditions and environments they put them in. They don't just do them all in the same, in the same swoop, right? So they will do like RNA synthesis in various different steps, even to get one nucleotide, right? And then same for the protein synthesis with amino acids. And those are in a different condition than what they do the RNA with. So yeah, we're going to get a little bit more into that.
Yeah, so it's crazy, all these different environments that each one of these pieces have to, possibly be made in. And it's crazy they say that UV radiation is used as an energy source for DNA and RNA when UV radiation causes thymine dimers and it has to be cut out in order for DNA to be replicated.
Yeah, so it's crazy. So they have to invoke this UV radiation. But at the same time, if prolonged to UV light too long, then the DNA is going to denature. So very interesting. All right, so I was trying to find this current status of abiogenesis, and basically I came up with this summary right here. So between the edges of a hydrothermal vent, ocean water, and a volcanic rock pores formed nature's chemical laboratories. Each pore offered a unique microenvironment, some stockpiling amino acids, others stockpiling nucleotides, while a few fostered the assembly of RNA strands. Lipid components delivered by meteorites crashing into the Earth provided raw material for the primitive membranes. Lightning strikes while UV light is radiating the shallow pools, driving critical reactions. Meteorites crash near these pools, delivering essential elements and providing energy for certain chemical reactions to occur, like to make nucleotides or RNA. And then you have this clay surface, which acted as a molecular assembly line, aligning and concentrating molecules in a pore of rock near a hydrothermal vent, while magnetic minerals align them into just the right configurations. Furthermore, relentless wet and dry cycles in these pools alternately dissolved and concentrated molecules, forcing them to link into more complex forms. Eventually, RNA migrated from the shallow pools to the hydrothermal vents, and tiny lipid bubbles encapsulated the RNA that emerged, while being able to still hold the contents and transfer materials from inside to outside of the cell without breaking down, and began migrating between these environments, scavenging components and energy wherever they could. Through this interplay of hydrothermal vents, meteorites, lightning, and UV light, and drying pools, the first cell was born. Right.
I'm sorry. That's the most retarded conspiracy theory I've ever heard in my life.
Are you kidding?
That is the most absurd story that anyone has ever told me about anything.
This scenario here has the makings for an immensely great Disney movie. I see cartoon characters coming out of this left and right.
Yeah, so like different parts of the cell, different molecules are going to require different environments. So that's why they have to invoke one environment for one thing and another environment for another thing. But whenever you put it all together, it just becomes this. And that's what we got.
Look at all the contradicting scenarios that it has to be high, low, top, bottom, left and
Yeah.
right, top and down, all at the same time, and just happen to, but at the same time, cause itself to permeate. All of a sudden, poof, it caused itself to exist.
Yep. In
Have
this
wet-
order.
Something like that.
This is
Something.
amazing. Thank you,
Yeah.
Taylor.
Yeah, no problem. All right, so I'm going to go over a couple different paradoxes. So the compatibility paradox is another example from all of these different contradicting scenarios that we'd have to have, right? With the temperature, UV light, etc. All right, so one example is the Maillard reaction, which is where amino acids can react with sugars like ribose, a component of RNA, in a Maillard type reaction, which could deplete the available ribose for RNA synthesis. So if you have these two together, they can react with each other and basically make them null and void. They're not going to be used anymore.
This reaction works very well. It's used for testing for sugars in urine.
Yeah.
Yeah.
So you can just see like how even though these different components need to be next to each other for life to form, they cannot cohabitate. And this is why the cell has these like these compartments, right? That'll separate these things. But whenever you don't have that, you have to invoke other means to separate them. And that's kind of where the craziness comes in with the trying to make a one good abiogenesis theory, because you have to have all these different microenvironments. So remember earlier how I was talking about how the cell is complex? Well, it doesn't just lose the complexity. The complexity issue is just going to be pushed onto the environment. That's why now, instead of the cell being complex to regulate for these things, we have a complex environmental scenario for the origin of life. And it's science, right? All right. So here's another paradox, the water paradox. So water is indispensable for life because it facilitates the chemical reactions and supports the folding of biomolecules like proteins and RNA. However, water also breaks down essential molecules like nucleic acids and proteins through hydrolysis, making it challenging for these molecules to form or persist in aqueous environments. So this is kind of why they have to have these like wet and dry cycles, because you have paradoxes like this that exist. All right, so here's the DNA protein paradox. So modern cells require proteins to replicate DNA and RNA, but proteins themselves are encoded by these genetic materials. So this creates a circular dependency where the components necessary for life rely on each other to exist, complicating the understanding of how life got here. So you can see DNA is used for transcription of RNA and used for translation of proteins. So the proteins are used to produce DNA. DNA is used to produce proteins. Proteins is used to produce RNA, and RNA as rRNA and tRNA are used to produce proteins. So it just kind of goes in circles here, right? And these would have to like come about at the same time, right? Because one of them without the other would be useless. If you just had a DNA strand but didn't have any like machinery to translate it or make it useful, it's just meaningless. So yeah, that complicates the issue. We also have the Huygens paradox, which is that we can't have long strands of DNA unless it can repair itself. And they can't repair itself without long strands of DNA. So this is very interesting. And this was a excerpt posted from Nature. It said he posed the paradox that without error correcting enzymes, the length of a nucleic acid would be limited in larger molecules. Mutations would destroy the information contents of subsequent generations. But this maximum size or error threshold was too small to encode an error-correcting enzyme. So Huygens paradox still challenges theoretical biologists. And then end quote. That's so funny that they said theoretical biologists. I'm assuming they're referencing the original life researchers. So anyway, here's a picture of him over here. Manfred Huygens.
It's kind of like we have theoretical physicists. You have theoretical biologists, right?
Right. I didn't even know that existed. But yeah, he's giving a nice little thumbs up. He's like, how you like that? Yeah, try to figure that one out. All right, so and this little schematic kind of goes over it up here too. Okay, then we have the ribozyme paradox. Okay, so let me just say this. So the DNA paradox, the scientists well aware that this is an issue, right? So in order to solve this problem, they came up with the RNA first world. Okay, so in this theory, basically you'll have RNA that comes first, and then from there, DNA and proteins will come later. So then they propose that you could have the RNA basically fold into this ribozyme that can like self-replicate, right? So that's their explanation or attempted explanation for the chicken and the egg problem with DNA and proteins. Okay, so and this ribozyme actually has paradoxes within itself, right? So there's paradoxes within this explanation. So in order for a ribozyme to be functional, it must be stably folded into a complex structure, much like a modern day enzyme. However, in the absence of set-aside genome, this same ribozyme would also need to serve as a template for its own replication. Templates cannot have complex, stable folding if they are to be accessible to free nucleotides and an RNA primer. Clearly, the same RNA cannot meet these two mutually exclusive requirements simultaneously. In the RNA world hypothesis, ribozymes necessarily must have these dual roles. They must have catalytic activity while simultaneously being able to act as a template for replication. So catalytic activity would require stable folding into complex structures, whereas its role as a template requires unstable, relaxed folding RNA. So it cannot be folded, right? But it has to be folded. So that's kind of like the paradox here. So ribozymes must carry their own genome to be replicated. They must have templating activity, which requires unstable folding. Ribozymes also must possess catalytic activity, which requires stable folding. So the catalytic activity is incompatible with the templating activity. And then also the stable folding is not compatible, obviously, with the unstable folding. So you can't have this ribozyme basically self-replicate. So you'd have to have another ribozyme at the same time. So it's crazy.
I'll be addressing some of these like self-reproducing ribozymes later.
Okay, cool. Yeah, so there's just like paradoxes on paradoxes here. Okay, then we also have the circular synthesis paradox, right? So over here is the citric acid cycle. So in the circular synthesis paradox, you need things like ATP to make ATP. So in order to make ATP, which is the energy of the cell, you need 6 ATP. So it's crazy. And then you also need NAD to make NAD. You need tetrahydrofolate to make tetrahydrofolate. Coenzyme A is needed to make coenzyme A. And then you need FAD to make FAD. So like you have all these like circular synthesis reactions and like which one comes first, you know? So yeah. Then you also have the membrane paradox. So the membrane formation paradox acknowledges that the cell membrane can only be synthesized from pre-existing membranes. So what it's showing right here in B is, as an extension, the organelle formation paradox states that membrane and capsule organelles can only be produced from pre-existing organelles. And then C is showing the protein phospholipid paradox, which demonstrates that the protein composition in an organelle is dictated by the organelle's phospholipid composition, but the phospholipid composition is dictated by the protein composition. So it's just like constantly going in circles here. So like, how did these things come about? You know, they must have had to come about simultaneously, and that would obviously invoke some crazy radical like interference, right? So did you have anything to say on this?
It's just interesting how like all cells like they share their membranes and stuff. So no membrane just comes from nowhere.
Mhm.
And then like there's membranes. We way back towards the beginning we talked about the endoplasmic reticulum and how cells get rid of product and stuff. These membranes are all like connected and folded together, and it's like really more intricate than most people realize.
Yeah, definitely need some intervention for that. Okay, and then after this slide, I believe is your slides.
Okay.
Okay, so here's the tRNA paradox. So tRNAs are essential for protein synthesis, yet their processing and modification requires proteins. This creates a chicken and the egg problem in abiogenesis scenarios, where neither system can coexist without the other. Okay, so yeah, over here you can see DNA is used for transcription RNA, which is used for translation of proteins. Proteins are used to make DNA, and so on and so forth. So yeah, did you have a few slides? Yes.
Mhm.
Okay.
All right, so we're going to talk about like transfer RNA, and I know we're jumping right into this, but you see this this red ball here. This is the like That's the ribosome. Okay. And so you need tRNA, and that's these purple lines right here. And what we're going to be doing is like connecting mRNA, which is what we saw back in the YouTube video about splicing. This is the mRNA that's coming out of the nucleus, going into the cytoplasm. And then this little diagram here is showing how there's going to be multiple ribosomes connecting to that mRNA and making completed proteins. So these purple lines, the tRNA, is going to be translating between mRNA and amino acids. So the role, I kind of explained it, right? It's going to be reading the genetic code, attaching amino acids together in order to build protein. All right, so just a quick look at its structure here. It has this anti-codon region, which makes it specific for the amino acid codon, the mRNA code that it'll be reading. Okay, and then it has these arms right here, and we're not really even getting into the detail of this, but these have to be designed in a particular way in order for it to fold and to function correctly in the ribosome. All right, so what is this why is it called translation anyways? Well, because there's two very specific chemical languages using different spelling, or orthography, and a system of units, its morphology. These have to be matched up correctly. And so there's 64 possible codons of mRNA, and it has to be matched for the 20 different unique amino acids. And you can use this wheel right here whenever you're trying to predict like what mRNA sequence is like going to create which amino acids. You just ACU, for example. And
Yeah, so they basically speak like two different languages.
Yeah, so what I'm saying is there's two complete different languages.
And you have to have a mediator between them.
Yeah, and we saw earlier how they were made differently. Each component of the languages are made
completely differently,
organized differently. It's really great. So we talked about this transfer RNA. How does it get that specific amino acid? Well, this other process called tRNA charging. This is a really important process here, and we're done by this preformed protein, right? We're talking about this protein has to be there, aminoacyl tRNA synthetase, and it uses, and there's 20 unique ones, right? It uses its one unique enzyme to bind each correct amino acid onto each specific tRNA, called charging, after which it scrutinizes or judges the amino acid before and after the binding. And if it's not right, it'll cut it off and try to match the correct one. If this doesn't work correctly, you can have a disease like Charcot-Marie-Tooth disease. And, so we talked about how there has to be this preformed enzyme in order to bind the amino acid to the top of the tRNA. Next is the ribosome. Okay, and you may have seen a picture like this before in your biology textbook where the process is actually going on. But I just want to mention this thing is far more complex than people realize. This thing is made up of two very large RNAs and over 50 preformed proteins, all of which have to be made with transcription.
At the same time, they have to be there. Yeah, but how would they get there?
Exactly.
That's the problem.
And then so there's three sites in the ribosome. There's the A, the P, and the E site. A is where the charged amino acid tRNA binds, and then it goes to the P site called the elongation site, and it's where the amino acid goes through a process, forms the peptide bond, and builds like a chain of amino acids. And there's the E site where the tRNA just kind of rests before it goes back and restarts this whole process over again. And I just want to show you this is another good diagram here, kind of demonstrating how that happens. So we start here, it moves over, and then the second one pops in, makes a peptide bond, and then it continues on making a protein. And in case you just thought, oh, well, that's it, I included some more details here of how transcription actually occurs. And there's all these other little helper proteins like release factors and binding factors to make all this work correctly. And then now I wanted to address something that we talked about with self-reproducing tRNAs. This was a paper that tried to claim that this tRNA sequence assembled itself. And you know how the titles can always be super misleading.
Yeah, never just look at the title. You have to go read the materials and methods. There's so many cases like, oh, we created this in the lab. And then like, I'm like, oh, let me see. And then basically they have all these like subunits that were already formed. And then they just had some fancy chemical reaction. They're saying, oh yeah, we just created it. Like, it's crazy. But that happens all the time.
And so while the whole body of the paper is trying to discuss how this could have happened in early Earth conditions, they're using a thermal cycler or a PCR machine, which will go from 90 C to 50 C every 20 minutes. and it will do that like 30 or 40 times.
Yeah.
And this was used in order to create the thermal oscillations needed to reproduce the DNA. That's how that's how a PCR machine works. I
Right.
described it here. But these pre-synthesized tRNAs were designed specifically to possess a specific structure for replication, and they tried to remove anything that would avoid that from happening, right? And so that's totally not how it would arise in a prebiotic world. Additionally, these specific sequences, maximized replication efficiency and minimize non-specific amplification.
Yeah, and the whole thing with this is like this actually gives proof for an intelligent designer. Like if you have to do all these like subsets of an experiment and highly regulate them, you're literally intervening right there and showing like in these like prebiotic synthesis experiments that like you need somebody to intervene, give it the right pH, cut it off at the right time, separate it, et cetera, et cetera.
Exactly.
So tell them how you got this paper, like basically what happened.
What do you mean?
I was he was doing the tRNA.
Yeah, okay. Just like
okay.
studying tRNAs and like trying to look at them and stuff, and then we came across this.
Then I was like, well, you have to make sure you look at any rebuttals, like before you
Oh,
make a
definitely.
claim. You have to at least address them. So
Yeah, and then the error rate for this was like 10%.
but they
which is
didn't even
crazy
actually
high.
create a tRNA from scratch. Like that's like
Yeah,
people
all
might
this is
think-
lab ordered.
People might think that, but didn't they already use a tRNA and they just modified the genes?
They designed what you can do is actually order specific sequences of a genome and then have it, shipped to you.
So
And
they
that's what
they already had it designed, and then they just put it through a PCR and basically got the protein replicated. So either way, like they're not just happening from scratch.
For the benefit of those playing along, PCR, is that the same acronym that we've been talking about in the public for 4 years? That's polymerase chain reaction?
Yep.
Yep.
I thought so. Cool, thanks.
So they didn't create a tRNA from scratch or whatever they were trying to claim, but you have to just look at the materials and methods to see what they actually did.
And then even this thing they did made, it was like error rate of 10%, which is like crazy high. Like our error rate has to be like less than like 1 in a million,
And then this paper doesn't even solve the problem of, first of all, how it arose, where did it get the sequence to arise, and then how would it be mediating between these two very different conditions needed for RNAs and proteins to be in the same area. So how would it be mediating? And then the biggest problem is how did it translate between them? They're two completely different languages. Does this paper even tackle that translation problem between them?
No, this is just trying to claim that the thing made itself.
Yeah, it's crazy.
You want me to continue about
But they already used the template. Crazy. All right, so here is a brief side note. The phosphate problem, just one of the many problems, right? So bioavailable phosphate was required for both energy production and as the DNA's backbone for a cell. However, phosphorus-containing minerals on early Earth, such as apatite, were highly insoluble and basically bound up into rock. And so this poses a huge challenge for the, creation of life, because you need this phosphate that's bioavailable in order to have chemical reactions occur, right? And then so it was so funny, like NASA basically came out with a paper trying to solve this problem, and they were saying, oh, early life forms could have incorporated arsenic into its DNA. And then after other scientists basically looked at it and tried to reproduce it, they found out NASA, like the whole experiment was just flawed, and it was really bad. So here's a little excerpt right here. It says scientists say NASA's new arsenic form of life was untrue. So they basically got called out by the scientific community, and yeah.
This person got fired.
Is that the same article I saw before where critiquing it, they just finally came out afterwards analyzing it, saying that only 10 of the 14 components were actually found? So it really wasn't.
Maybe so. They definitely had
form of life?
to. They had to definitely walk this back. It was like a big PR
Well,
problem.
yeah. So NASA was basically saying that arsenic could form the DNA backbone. But whenever scientists tried to reproduce this, right, and tried to see if that could actually happen, it did not happen. So they basically just made a false claim and got called out by it. But there might be other experiments I'm not aware of that you're referencing. Yeah. So this is just the phosphate problem. All right. And then the time paradox, right? So will time solve their problems? Like with enough time, surely it'll all work, right? So complex organic molecules require time to accumulate and become useful through these random chemical processes. However, time is the very thing that will degrade the organic materials. So it's paradoxical here. You need time to build up these biopolymers. However, over too much time, they will begin to degrade. So yeah.
I love this one because this is one you hear from so many people, like you never know what
With enough
happens,
time.
billions and billions of years, but like it all like showed me something that's
Doesn't
lasted.
matter. Like it at time can hurt them in many situations. So
Mhm.
yeah, here's just
Yeah.
abiogenesis being posted right now.
Yeah, things
Little-
fossilize in billions of years, according to them, right?
I don't know. All right, so this was a funny paper. So it says the RNA world hypothesis, the worst theory of early evolution life, except for all of the others, right? So this is really funny. Like RNA world is basically a bad theory, but all the other ones suck just as bad. So yeah, this is hilarious. So they acknowledge that there's problems with every theory, and this is why many of them can't even come together on a theory. Some of them will say protein first, metabolism first, RNA first. And please keep in mind, this is still step one. We can't even figure out like what came first. And there's problems with each of them. That's why there's competing theories. If there wasn't problems, there wouldn't need competing theories. But there is. So anyway. All right. Here was a British origin of life researcher, John Sutherland, and he basically conceded to the lack of progress in explaining a naturalistic origin of life. Right? So they have all these reactions that need to occur. And then he basically says down here, look, the current state of the field, right? Right here at the very beginning. This is hilarious. Anyway, so he's conceding that they haven't made much progress. And this is a prominent origin of life researcher, by the way. I came across some of his papers when doing research. All right, so here's a statement by him. Do you want to read it? Can you read
Yeah.
it? Thank you.
Understanding how life on Earth might have originated is the major goal of origins of life chemistry. To proceed from simple feedstock molecules and energy sources to a living system requires extensive synthesis and coordinated assembly in order to occur over numerous steps, which are governed only by environmental factors and inherent chemical reactivity. Demonstrating such a process in the laboratory would show how life can start from the inanimate. If the starting materials were irrefutably primordial, and the end result happened to bear an uncanny resemblance to extant biology for what turned out to be purely chemical reasons, albeit elegant, subtle ones, then it could be a recapitulation of the ways that natural life originated. We are not yet close to achieving this end.
So they're basically saying they're not even close to this happening.
Yeah.
Yeah. So anyway, so that was basically the gist of the quote. And then he just says, but recent results suggest that we may have nearly finished the first phase, right? So they're like on phase one.
Oh yeah.
And he's admitting that too. So they have these original life researchers actually admitting it. And then here's another excerpt. The prebiotic synthesis of building blocks to which we have devoted so much of our time only corresponds to a small increase in the complexity of the system and to no increase in its aliveness. And this is a humbling thought, he said. So there was actually in the conference of Original Life, they were trying to figure out, well, what actually constitutes being alive? Like, remember like the characteristics of life that I gave you guys earlier? They have to have all these different functions and characteristics, being able to respond to their environment, being able to utilize energy, etc., and reproduction, etc. So basically, like, where do they start? When do they say that they can reach life? Because they're not even on step one. Like, they haven't even completed step one of how to get like a viable polymer that can function from prebiotic conditions and inorganic materials. They just haven't done it. And yeah, it's very evident when looking into their research, like how many things they have to actually invoke and just to do small subsets of experiments. Like just to make one nucleotide, they had to do multiple different types of experiments. And it's just like they're doing these tiny little sub pieces and they're doing them all in different conditions. So if you have to do them all in different conditions, like how is it ever going to come about in nature when they're all supposed to be in the same area, right? So it's just, they're just having a hard time moving on. And for obvious reasons. So I wasn't finished. I was almost finished. Okay, so around 100 years ago, abiogenesis was proposed, and we're still not even on step one, right? We still haven't completed step one. We have yet to successfully create a functional protein from inorganic molecules under prebiotic conditions. So, like I said, they'll do these like experiments like where they'll already use amino acids and then they'll get them to bind in these like crazy conditions that are highly regulated, but they're not starting from inorganic materials, starting from scratch and getting these amino acids to form and then turn into a protein like from scratch. They're just It's just, not
Not
there.
even when using lightning. Not even when using lightning and UV at the same time.
Well, yeah. Well, see, remember that tar that was produced? Like, so what are they going to do? Take that tar and try to get it to form a protein? Like, come on, this is the problem. So then for the next step, they'll say, okay, well, we've made amino acids, so we can move on from there. So now we just need to get them to bond. So they'll take these highly regulated, already chiral amino acids or whatever, and then get them to bond, and they think that they made progress. But notice how they're starting from scratch every time. They're not just starting from the beginning and getting the end. And this is just for one protein.
Yeah, like jumping over the roadblocks.
Yeah, like they have the Miller-Urey for the amino acids, and then they'll take amino acids and then go from there.
Okay, but what if they invoke a third catalyst? What if they take the lightning and the UV and they do that spark during a solar eclipse?
You never know. That might be the next thing to happen. I don't know. I just by looking at the conditions that each of these parts form in, I don't know how they would do them simultaneously. Like there's critical issues from starting from inorganic materials to get both a functional RNA and a protein. It just hasn't happened, Yeah. So
Can I ask
scientists have.
a specific question on this slide?
Yeah, go ahead.
Yeah, you two say stuff like a living system requires specific synthesis and coordinated assembly to proceed from simple feedstock molecules, as if time doesn't solve all issues. Why do you two philosophically choose to rule out infinite time as a sufficient answer?
Because
We'll see. We addressed the time problem.
it wouldn't even help.
Have you ever seen anything last for infinity?
Yeah, and then especially not these organic molecules, which degrade over time.
Yeah.
So,
I got a good slide coming up about philosophical arguments.
Right.
Touché.
So
And that takes us back to fossilization.
exactly. So then here's a fun fact. No living cell has ever been created in the lab from non-living components. And they actually did a survey, and many people actually thought that they created a living cell like from scratch, and that's just not there. They'll use like an already living cell and then put like a new DNA strand in it and then act like they created the cell, but they're using an already living cell. Or they'll get like this self-replicating ribozyme, but they already used an RNA polymerase that they just, modified and then said, yeah, we made a self-replicating RNA, but they're using stuff from already living cells. And then in another experiment where they were saying they created RNA, basically they used the primers of an RNA, right? They already put the primers in there in the nucleotides, and they're like, oh, we got something from it. Like basically from one of those strands, they had some like some folding occur, and then they said, oh, well, these must be, replicating themselves or something. But in all these cases, like they're using primers, they're using RNA polymerase and highly regulated environments, which actually proves intelligent design to have so much regulation in any of these experiments. So, yeah. All right. So I just wanted to remind you guys, like if, we think that they're getting close to life, from these experiments, what a simple-celled organism would actually have, to be able to perform, right? I kind of mentioned these earlier, but they have to be able to have energy production, storage, structure, transport. They have to grow. They have to reproduce. And you could just see like where we're at in abiogenesis research is nowhere close to this. And that's why the abiogenesis researchers said like we're still here on step one. And it's actually not even like they're getting closer to the answer. It's actually like the more we're learning about the cell, like the further away we're getting from the answers. Like remember earlier I said that the cells they thought were just like these this protoplasm and this nucleus, and that's basically they thought it was just simple, right? And that's all they knew at the time. And then only after we had electron microscopes could we see like the complexity, right? So, but now the more we learn about it, the harder it becomes to answer abiogenesis, and that's just continuing on. So the more we learn, the harder it's getting. We're not getting much closer, in my opinion, to solving this. Anyway. All right, and here you go, Dan.
Yeah, so I wanted to address like some philosophical basis for detecting design. So it's just like when you're inferring design on something, it's a rational scientific process based on identifying features that are known to arise from intelligent agents, right? And it's a really good quote from Stephen Meyer. He says, when there's two things that are specific and complex, that's a type of information that only arises from a mind. And that's what we have inside of living cells. So, just as it would be crazy to say that, Mount Rushmore came about by natural processes and erosion, obviously, right? So too would it just say it's just an accident how, T cells differentiate themselves between T helper 1 and 2, and this is even just a simplified model. And so only intelligent agents are responsible for complex information, right? So in the same way we have the genetic code, so too do we have the binary code that we designed, right? And then furthermore, there's like integrated circuits and hierarchical information
in DNA, right?
Yeah, in
In the
DNA.
DNA
Yeah, so that's what I'm saying here. There's actually many examples of it. So just like this computer circuit here, we have there's hierarchical systems and circuits, right? So DNA, genes, chromosomes to genome. But then on a larger scale, you have organelles, molecules, organelles, and cells. And these cells make tissue, organs, organ systems, organism. So too you have neurons, neural circuits, brain regions, and the whole central nervous system. And then this is actually super cool here. This is actually a really good map of biochemistry, right? And so remember we talked about the citrus acid cycle earlier. That's this right here. And you have different forms of your metabolism, both feeding into it and then going out of it.
So this is for energy production in the cell, right?
This is for a lot of different things.
Metabolism, right? Primarily, right?
Yes, somewhere on here is going to describe where our cells make all the mRNA and DNA and sugars and stuff. I just want to show you that if you think that this is complex,
look at
just
that.
look inside.
And we haven't even discovered it all. So it might be more complex. But this is just like looking at metabolism. You can see like the circuits that are already encoded into the cell. So yeah,
absolutely. And so in a similar manner, you have this happening in things that we talk about every day, right? And I just wanted to mention a slide I'm learning from my upcoming test, how, vitamin D, has to go through multiple different organs, and then its effects, affect all these other different organs, and they feed back on themselves, and all like in a really nice regulated way.
Yeah.
And so
this might be mine.
Yeah, this is yours. Go ahead. Like some positive cases for intelligent design. They're complex and specific, like we just addressed. There's irreducible complexity, like we addressed in the cilia. How if you take a piece out, it doesn't function. It's purposeful. It's fine tuned. It's information rich, and it's non-random. It has to meet these specifications. It has to be adaptable, right? Because it has to adapt to our environment. Like every time you affect the body in some kind of way, like it's going to respond in a different kind of way, usually to protect itself. And it has to create new information, right? To be adaptable.
The creation of new information. Yeah. These are all indications of intelligent design that we can actually see, in the cell. And so here's a cool little depiction of some of the machines in the cell, right? So you got this ATP synthetase, right? And then you have this flagellum, and this actually resembles a rotary motor. All right, so specified functional complexity. So here was a statement by Charles Darwin on the origin of species. He said, if it could be demonstrated that any complex organ system which could not have possibly been formed by numerous successive slight modifications, my theory would absolutely break down. Right? So basically, if we find irreducible complexity, his theory would break down, right? And then so we actually look in the cell and we see, wow, I think we created something like this, right? We created rotary motors. It's not like what's happening here. Like that's crazy. So we actually designed rotary motors, and later we looked in the cell and saw rotary motors there. So it's absolutely crazy. So here's some of the parts of a flagellum.
This one's way cooler. It goes like 100,000 RPM and like is near 100% energy efficiency.
Yeah, so it's actually better than our motors that we created. Like, yeah, we were definitely outdone with this. So
you were going to talk about the parts, maybe?
I was just basically trying to show how many different parts there are.
Oh yeah.
And then there was like pro evolutionists trying to say, oh well, we found a different type of flagellum, and that means that it can function with less parts. But actually, that different type of flagellum, they said, came from a different like origin point, right? And that would imply that basically they had a split off, right? And they each came up with their own, rotary motor. And I feel like this is more evidence for intelligent design. So Yeah, it's not even like that there's a flagellum that it's less complex. The flagellum that they say is less complex in this particular bacteria actually needs all of the parts as well to function. And if one of their parts was missing, it would have it would be detrimental. But either way, we can see indications of an intelligent designer here in these motors. Because if there were pieces missing, it would not work. And by Charles Darwin's own words, his theory should be broken down. And then there's other cases like in the ATP synthase, and there's a lot of similarities with man-made motors, right? So here's some pictures of the ATP synthase. Basically, they have this proton pump, and as the protons are moving through it, it's basically spinning this little, would that be a rotor?
Mm-hmm.
And
Yep.
then energy is being created. And here's some pictures of it from the electron microscope.
Like the way this works, like you might see in your biology books as well, like that there's a proton gradient, a bunch of like protons on this side. They flow through here, turn this, essentially like a water wheel, a turbine, and then it turns this in order to make ATP. And there's like a whole nother problem to this,
Yeah.
that, we don't even
A lot of-
understand how it works really.
Yeah, there's a lot to learn with it. It's amazing. Some biologists called it like the most amazing like machine that's ever been created, and it has like near perfect efficiency. So, and this is actually really slowed down. Like if you were to see like what this would look like in real life, it'd be like zooming. Like it's amazing. So, okay, let me read some of the similarities. So, ATP synthase and man-made motors share key similarities. Both will utilize the rotational mechanisms with a central shaft rotating within a stationary housing, resembling the rotor and stator design in electric motors. They generate torque to perform work and are modular in structure. They're also adaptable, such as that ATP synthase can operate in reverse. Additionally, they convert energy with remarkable efficiency, except ATP synthase achieves a near 100% efficiency. So you can just see like the, amazingness of this motor. And, without all of these parts working together, like what would be the purpose of evolving to create it? It would essentially have no function for so long, and it, would have no output. So if any of these parts were missing, it would not work. Yeah. And then so also we can see indications of an intelligent designer in DNA. Okay. So we have complex specific information. So basically, in an intelligent design, if we were to say, hey, I wonder if this is intelligently designed, we'd look for these key features, right? Complex specific information. So DNA's organized code acts like a digital instruction, right? Also irreducible complexity. DNA replication needs all parts at once. Purposeful arrangement. DNA bases are arranged to code for proteins similar to a program. It's fine-tuned. DNA structure is precisely suited for storing and copying information. Information-rich features. DNA holds detailed instructions for the cell. In specific complexity, DNA's coding regions are complex but match the functional needs, of the DNA. So, there's a lot of different indications of an intelligent designer just within DNA. And that's kind of what I talk about here in this slide. So we see with intelligently designed products, we can see integrated systems or an informational hierarchy. And we only see these in intelligently designed products, right? If something has integrated systems and informational hierarchy, these are indications of an intelligent designer, right? And so DNA has them with enzymes, right? When the DNA replication, transcription, translation, DNA repair, and gene regulation. There's a lot of systems that are already integrated in it. And earlier you saw one of those integrated circuit boards, like with the metabolism, for example. You also have informational hierarchy, right? So you got the base pairs, they store genetic information. Then you have these codons that code for amino acids in pairs of 3. You have these genes and segments of DNA that encode proteins, and you have these chromosomes that highly organizes the DNA into higher structures. All right, in evidence for intelligent design, we have is this your slide?
Mm-hmm.
Okay.
Yeah, so we talked about this a lot, but we're just kind of addressing some of these things on the final, that all these things work together in a tightly integrated network. Each component's dependent on itself. That's what irreducible complexity is all about. We talked about the ribosome, but it's a molecular machine needing 50 different proteins that have to already be made in advance in order to make a protein.
Wow. So you have 50 proteins that need to be made in advance for one.
If any one of them are missing, it does not function.
Wow. So ribosomes, if you saw earlier on my chart about the organelles, they're actually responsible for creating proteins. And those require 50 proteins. Think about it. Crazy.
So
Yeah.
that supports intelligent design. It has to be super precise. What the specific codons, like from the mRNA to the amino acids, and that's what the tRNA's job was for. And so then I have this is just a couple questions here. We can make a bunch. Some challenges to abiogenesis, right? How did RNA molecules form and stabilize in prebiotic conditions? How did the genetic code of DNA, mRNA, tRNA, and amino acids arise? And how did tRNA's ability to recognize codons and amino acids emerge, right? Where did it learn this language?
Yeah.
2 different languages. Yeah.
2 different, and you can like think about it, like if somebody's just sitting there staring at a computer code all day, right? They don't have any translator between you and the computer code. So like they're just sitting here staring at a code of ones and zeros. How long will it take before they can actually translate that? There has to be some mediator, and that's what the tRNA does. But the tRNA itself is amazing. Like how would it come about? And so it's just crazy. Yeah.
Exactly. And then that whole like biochemistry page of your metabolism,
Yeah.
where did that integrated circuit come from?
And how would it know to move forward if you have no feedback, if it wasn't actually doing anything?
Yeah.
And it wouldn't be able to do anything unless you had the whole circuit already there.
Exactly. It can't
Yeah, it's
feedback.
crazy. All right, so I'm finishing up here. Is abiogenesis a viable theory, right? So theories need to have a coherent explanation. Like, what is the theory? Is it RNA first? Is it DNA first? Protein first? Like, what is the theory? Okay. So yeah, abiogenesis just simply does not have this. And that could be evidence against it, right? So also, I see them appeal to the future a lot, right? Which is a logical fallacy where somebody's claiming that the argument or belief will be proven at some unspecified point in the future. So they say, oh, well, with time and more research, we will surely figure it out. But it seems like we're actually going in the opposite direction, because at first we just had the pool and the soup that Miller Urey worked with, struck by lightning. Now we have all these different environments that we have to have now that we learn about the complexity of the cell. So we need the porous rocks, we need the volcanic ash, we need the crystals for chirality, etc. And then personal incredulity, when someone dismisses a claim or argument simply because they find it difficult to understand or believe. You can't just say, oh, well, I don't understand intelligent design, I can't wrap my head around it, therefore it just can't be true. We have to have a materialistic explanation. That's just fallacious. All right, and then is intelligent design viable, right? So think about Occam's razor. It prefers explanations with fewer assumptions. Think about all the assumptions that we went over earlier for the first life to form, okay? And I can't even count how many of them there were. And intelligent design only has one. It only has one postulate, one abstract postulate. And so based on Occam's razor, the fewer assumptions would be intelligent design. And then also, as I mentioned earlier, the prebiotic experiments actually point to an intelligent designer. In these experiments that they're doing to try to recreate abiogenesis and say how life could have arisen from non-living matter, they're having to heavily regulate them. They're having to basically go in and have stop points and start points and highly, isolated amino acids of all the same chirality in some cases. And they're basically just tightly regulating to make this one reaction happen. And they're intervening. They're like for the lipid synthesis, they had to basically centrifuge it and then like put it together in order for it to create this like lipid membrane. I don't even know if I'd consider it that, but whatever. They have to like concentrate it together, and they're like, this would have formed very low concentrations, but we're just going to centrifuge it and fix all that, right? So anyway, the heavy intervention in these experiments is really just showing like evidence for an intelligent designer or something that was intervening, right, at the very least. And then intelligent design is not just the God of the gaps. It is based on what we know and what we can see. So what we know is that information that has integrated systems and informational hierarchy and integrated circuits, this type of information comes from an intelligent designer. So we can just say, okay, well, we know where this type of information comes from, and we see it in the DNA code. So what can we infer? And can we create this type of information by just random number generators, for example? Can we create it just by random nucleotide sequences or amino acid sequences? Have they been able to form something functional? Like, so we can actually do experimental tests with this and see, like, how does this information come about? How does it, how would it basically form, right? So we can see that indicates intelligent designer, right? And then we can observe attributes of something that is intelligently designed, and make inferences from there. So it's not really God of the gaps. It's based on what we do know by looking at nature and making those inferences. Not about a gap, is what I'm saying. And so that is about it. And so I would like to give acknowledgments to Daniel, my beautiful husband, for helping me. Thank you to Alan for inviting me on, and to Sam, Peter, Andrew, and others, Galatrax, on EtherCause Discord for helping me with the organization process. And thank you to all of the EtherCause family for hosting me today. I've enjoyed it.
Thank
And
you.
if y'all
Thank
have any questions, let me know.
Dude, that was an awesome Prezi, Taylor. Thank
Guys
you
are
for that.
pros, professional presenters over here. Guess
Dude,
what?
you
So
guys
every
did
once a
great.
month, we're going to expect a new topic, right? We'll get back to you with next month or whatever.
Yeah,
Yeah,
that's fun.
that was excellent, Taylor. I thought
Thank you.
my
Bye.
yeah, I thought my coffee commercials were really outlandish stories, but honestly, these scientists, they
The
are
story of
getting a lot more
abiogenesis.
money. They are way more outlandish than my testimonials.
Yo,
Yeah.
you know what's funny is the whole theory, like the philosophy of science, modern, when they go from the Copernican principle to try and get away from humans as a central divine figure, right? We always talk about it in relation to cosmology, but we forget it permeates all of science. So biology, biochemistry, specifically, everything they're trying to do to make abiogenesis a reality is a direct result of the Copernican principle at play and the philosophy playing as science, so that people are like, well, we know we're not special, we're not created, we're not divine, so therefore it must have been this ridiculous set of circumstances that happened all at the same time, but in total opposite to each other, that then resulted in life just starting. Yeah, that makes total sense, right? Like that's how they got there from that philosophy, I think.
It's crazy. Yeah. Yeah, it's absolutely crazy.
Yeah,
Great
there was
presentation, Taylor. Just wanted to say before I have to go back on mute. Great presentation.
Thank you so
there
much.
was one honest scientist when people asked him, do you think that this universe has a creator, is there an intelligent creator? He said, yeah, well, it's like giving a monkey a typewriter, and that by sheer coincidence it types up an entire play of Shakespeare.
I was
So
gonna
he
say,
was saying, of course there's a creator.
can you go
Yeah,
back to that super complicated slide that you were showing, and we could pretend that a cyclone will cause that by accident or whatever?
yeah, it's just like look at it long enough, and maybe we can
Yeah.
happen.
Or with enough time.
Slide real quick of Stephen Myers, because you just mentioned something about the universe itself. And yeah, here. Okay, so Stephen Meyer, he has another book. It's called
God
The God
hypothesis?
Hypothesis.
Yeah.
Yeah, it's very cool. He's a physicist, and he talks about how like all these different factors, like all these different variables that describe the universe, have to be set within like 1 over like 10 to like the 50th power or more. That like if they were to adjust it just a little bit, like the universe wouldn't be the way it is. It
Right.
couldn't harbor life.
Yeah, and there's a lot of that too with like protein creation. I didn't even really get into the probability, but like the chances of a protein being created from an amino acid just by, randomness, like and the order actually produces something functional is like 10 to the 180th or something. And
Yeah.
like there's more atoms in the universe than that probability, right? So there's or less atoms in the universe than that. So
Exactly.
So there are cases in which they can say something is statistically impossible, and it's by the standards of, from what I recall, if there's if the chances are lower than the number of atoms in the universe, you can basically say it's impossible. And for some of these cases, it absolutely is. Like the chances of one protein coming about just by a random amino acid sequence is astronomically low.
Mhm.
Yeah. And what were you gonna show here? Just
I
the
just wanted to go back.
Can
to
you guys post that image in the chat?
Yeah, I sure can. Yeah.
so yeah. If anybody has any other questions, just let us know.
Super
enjoyed it.
impressed with both of you guys' knowledge, and also impressed with your inherent tandem presentation that you just so nicely handled.
I know. I think that's the first time we've done that, and I really enjoyed it. I hope to do it soon again.
Yeah,
It really came across really polished, like you guys do it all the time. So
Thank you.
Well, somebody said that it's good to have dialogue during it. So then I brought Dan on, and then I was telling him, hey, you should make some slides on tRNA. And then he basically made his own little segment. I was like, okay, we'll just do each of ours.
Yeah,
For sure.
I like how the dialogue was always you reading his slide. Yeah, it was good dialogue.
Who suggested there should be dialogue?
Sorry.
You're good.
No, I'm
Yeah.
happy that you guys did this. This was awesome. I thought you guys were really respectful to the current scientific philosophical bias. You know, whereas like the refutation is, yes, but I have a philosophical bias that tells me I need to appeal to possibility all the time.
Right?
Right.
And then you guys laid that out, right? And the possibility is not that possible. It's extremely improbable. So it's not just a minor field of possibility. It's a massive one. It's a problem. We have a
Exactly.
problem.
We have a problem. We do. And they're never gonna acknowledge it. There are original life researchers who have come out and basically said, you know, guys, we're getting nowhere. But many people will never say that because
Yo,
they're terrified of the alternative. What is the alternative? They, you know, what's the alternative, right? So they're just going to keep grasping at straws to try to make it work.
Taylor, what's this chart we're looking at?
This is happening inside you, and
This is
every
metabolism.
one of your cells. You know, and it goes. I mean,
Yeah.
it's just yeah. You know, like
Yeah.
this is.
Yo, what's up,
I'm sorry.
Cass? We can hear you.
Good night.
Oh, I think
Here-
Cass
here-
is coming on
okay.
to ask a question.
Ribose.
Crazy chart.
Like, these are making those nucleotides that we've been talking
Yeah,
about.
here's their nice little.
But you see here, look. You see these enzymes in blue right here? These things have to be there in order to make the nucleotide.
Right.
You know.
So chicken
Yeah.
and the egg problem right in there.
Exactly. And
And-
so creating an activated nucleotide was actually something after I was doing the research that I found was really hard for them to do. So they're like making these subsets of reactions just to make one nucleotide. Like they have not made a fully functional activated nucleotide from purely scratch in inorganic materials. From the papers I read, they were basically doing sub experiments, right? Where they're like making something in this condition. They said, okay, well, we've already made that. So we're going to start with that and then try to do this in a whole different experiment, a whole different setting and environment. Like they're not just like making it in like a one pot, you know, shot or whatever. So they're moving it and continuing to add on to the last experiment instead of just doing it all in one reaction. Because it's very difficult. You know, you could see the complexity of what's needed for these cells, and they're having to put that on the environment because the cell is not losing complexity. It's just having to push that over to the environment. And that's why they have to have all these crazy environmental situations. And this will help you understand the intricacies of the cell.
And I'm showing this specifically this part right here for a reason, because even the simplest life, you know, it uses glucose and goes through a process called glycolysis. This is considered, you know, the simplest metabolism right here. Like we do all this. This is cellular respiration over here. Okay, but the simple part, you could see from glucose, it goes all the way to pyruvate, and that's turned into lactic acid. You know, and you could see it's not very simple, right?
Not
And
simple at all.
All these things feed back on itself. It requires nucleotides in the form of ADP in order to even accept that energy through these chemical reactions. It needs specific enzymes to make it go one direction or the other, or it doesn't function. I mean, I can go on and on. You know,
and so another thing was the paradox of like ATP creation. ATP is basically our energy in the cell, and so in order to make one of these ATP, you need 6 more ATP in order to make it. It's like how you know how do they come about?
Dude, imagine those things that are so amazing that they can keep creating themselves over and over again by creating their own ingredients from which to create themselves, which they needed from the beginning. What magical creatures!
Yeah, it's crazy. I thought the funny. Oh, go ahead.
A quick question. I really found it fascinating with the circular, I think it was called circular synthesis paradox,
Yeah.
Where they so I know there's a lot of apologetics where, especially one of them where you need a long DNA to repair DNA, so they can't exist without the long DNA in the first place. Do they use like do they use any like apologetics that they have with the evolutions where oh well the ones that survived were long, so it allowed them to replicate, but I don't see how that could exist in the first place. Did they ever say that maybe DNA started without needing any repair? Any apologetics around that?
We don't see any DNA that doesn't need repair. A long strand of DNA has to be compacted and protected. Something you hardly ever see depicted over DNA. If you see just the helix, that's not how it looks. It's totally coated in proteins, proteins that need to be translated. Because if not, it's it's very breakable.
Yeah.
Sensitive. It'll get twisted and break. It
So they
can
wrap
fall
around
apart.
the DNA, and it's a helix, but they wraps
Mhm.
around histones, and
Yeah.
then basically is in a nice little tight. And I have a little depiction over here of what it actually looks like.
Even more
We're looking at it
specific.
live in a cell, right? So and that forms this chromatin, which is the DNA and protein complexes that kind of keeps it stable. And so in the middle right here, the chromatin is in pink, and then the microtubules are in green, and it's basically dividing the cell. And so, yeah, so DNA is not just exposed like that. And it's also very interesting. I want to show y'all something else really quick. Like the inconsistencies. Okay, so DNA, the next one. All right, so notice like for the synthesis of DNA and RNA synthesis, like they use the UV light, right? They used UV light. And I'm assuming that's because it helps the bonds form and it interacts between the bonds between one another. However, once you have that DNA strand, then you have to get it out of the UV light, right? Because like you said earlier, you get these thymine dimers, right? And it starts like heavily mutating. So they used UV light for some of these experiments to make like RNA, to try to form an RNA molecule. However, you would need to remove it from the UV light, and yeah, and then the proteins was a whole other thing that was in like a hydrothermal vent, which is like a deep sea hydrothermal vent. So that would be in a different condition. And I don't know if they've ever like mediated this, but one of the ways they're trying to mediate it is by saying they have basically these porous rocks by these hydrothermal vents where these like subsections of different environments are located or in a different pore or whatever. Because they would have to be separated for some of the reaction processes.
Yeah, it sounds like natural selections couldn't handle all that. Thank you for the answer to that question. Awesome. I think
Yeah.
Cassandra was trying to ask a question.
Oh,
Thank you.
no, guys. Oh, no, guys.
Are you there, Cass?
Maybe we should check on them.
Yeah.
Well, I really appreciated it. If nobody has any other questions.
Well,
Um,
I
Dude,
think
Taylor and Dr. Dan, excellent presentation. Been listening the whole time. Excellent.
Thank you so much. I was a little nervous, but I'm not gonna lie, but I still had fun. But I'm actually more nervous presenting here than I am at school for some reason.
The standards are higher here.
I can tell you.
I was gonna say
All
I
right.
learned a lot, and I remember the one requirement I said was that you must distill your knowledge onto me by me doing no effort, and I think you've definitely done that. So thank you.
I tried.
Absolutely.
Well, hope y'all got something out of it. I guess if we have no other questions, I'll just end the stream.
Yep, we can go ahead and wrap it up. Taylor, thank you. Dan, thank you. That presentation was awesome. And we look forward to future prezzies dismantling the current mainstream paradigm on how they think the chicken and egg situation resolves itself with no intervention from any sort of some authoritative creator influence. That definitely isn't someone else in a lab making the structures that they're going to later use in their experiments to act like it, started from scratch and spontaneously made itself from nothing, even though they literally pre-ordered it. guys, they filled out an order form for the structure they wanted. They got it mailed to them cryogenically, I'm assuming, thawed it out, and then was like, look, guys, I'm God. Science is dope.
Not unlike cosmology or whatever.
Liliana, there's extraterrestrials that are possibly responsible for our creation. Are they responsible for the disappearance of brushtail coffee?
Yep, they are threatening to abduct me again if I don't give them more coffee. So I ordered more coffee. They'll only take it from me because they honestly believe that I make this coffee in my kitchen and that somehow I'm the only one that can provide it to them. So please buy Brushtail coffee. And also, while we're at it, folks, please consider supporting our wonderful learned colleagues. If you're on Alan and Shane's YouTube, they should have a link for their Ko-Fi accounts. And the best way is to subscribe to them monthly to support them. So please consider supporting our wonderful teachers. Yep. Thank you, Taylor. Thank you, Dan.
Yeah, thank y'all.
Awesome, Lily. Thank you for your contractually obligated fulfillment there. Fantastic work, as always. So tomorrow, so Wednesday, we'll be doing TikTok raids, and then Thursday, we'll be doing a follow-up TikTok stream. And then Friday, of course, will hopefully be the return of Flat Earth Fridays. If not, we'll be doing another community space. And then Sunday will be Globebusters. See you guys during the rest of the week.